So these are really kind of scary different bacteria and bugs that are present, and this is a growing threat nationwide. When we look at antibiotic-resistant infections, we find that they are the third highest cause of death in the US. This was in 2010, with over 150,000 deaths due to multi-drug resistant organism infection in this country. Further, this increase in antibiotic-resistant infections leads to very high costs across the country, with treatment of antibiotic-resistant infections costing $55 billion in 2013 to the US economy, and a projected amount for $100 trillion to the global economy by 2050, which isn't that far in the future. So this is a rising concern, and it's definitely something that we need to continue to address and continue to fight globally. Further, while we know that antibiotic-resistant infections are increasing, so here we're looking at from the 1980s to the 2016, we see that antibiotic resistance is on the rise with the red arrow. However, the discovery of antibiotics is actually decreasing. So the weapons that we have to fight antibiotic-resistant organisms are decreasing while resistant bacteria are on the rise. So what we're getting is this perfect storm of antibiotic resistance evolving while antibiotics are decreasing and more infections are rising. So this leads to, well, what can we do about it? Well, we're going to dig a little bit deeper into where antibiotic resistant infections come from and the best places to fight them. So actually, you are mostly at risk for you're at most risk of getting an antibiotic-resistant infection when you are in the hospital. The place you go when you are sick to get better and to get medicine is actually a place that can be dangerous because there are a high prevalence of antibiotic resistance. And so the most antibiotic-resistant infections we see are from hospital-associated infections. Things that someone is in the hospital, they did not have the infection beforehand, but then while in the hospital stay, they get this hospital-associated infection. And we want to understand, well, there have been decades of recognizing that this is a thing, and decades of trying to fight and figure out better studies of fighting these antibiotic-resistant infections, these infections still happen, and we have to better understand where they're coming from and where we can actually make methods and strategies to remove these organisms. So here, I'm going to walk you through the typical setup and general thought process of where these antibiotic-resistant organisms come from. So first, you have a sick patient who has an infection, maybe it's not antibiotic resistant, and they are given antibiotics. And when they're given antibiotics to treat their infection, that is putting a selective pressure in their gut where they have millions of native bacteria to all of a sudden develop and evolve antibiotic resistance. So maybe antibiotic-resistant bacteria weren't there before, but now after the antibiotic treatment, the patient is getting healthier per se, but there are now antibiotic-resistant bacteria. And so from this colonized patient, we imagine there would be contamination of this bacteria, often from fecal contamination. So if the bacteria is in your gut, the most frequent way of it getting out is actually from fecal contamination that may end up on different equipment in the hospital, may end up on contaminated hands of staff and visitors who are interacting with the patient, and may end up on surfaces, handles, sinks, bed rails, and other communal surfaces in the hospital. So then from here, from these different places where it is spread, we can then imagine it might end up in a susceptible patient who has no infection, is in the hospital for completely other reasons, but they are now susceptible to these antibiotic-resistant organisms and can get an infection that is antibiotic resistant. And now we can imagine that this is a cycle. So now from here, this patient would be given antibiotics and might move back over here, and we circle through the loop again. So there are things that we can do to address stopping fecal contamination and address stopping oral acquisition, but what do we do about these in-between steps? What do we do about these reservoirs that are being established? Now, what do I mean by reservoir? When I say reservoir, I mean a place where bacteria are living and growing and can stay there for long periods. So not just potentially dropped and there for one day until someone wipes off the surface, but there for long periods and continuously being a source of antibiotic-resistant organisms over time. And we imagine that these reservoirs are being established on contaminated equipment and contaminated surfaces over time, and that staff and visitors and patients are then spreading it from these reservoirs to other patients. So now, this is the general hypothesis of what's happening. So what can we do to fight it? Well, currently there's a lot of different methods that are being used in the hospital to prevent the spread of antibiotic-resistant organisms. There's some universal protection, such as if staff, visitors, and patients are spreading it, then if we focus on hand decontamination, such as hand sanitizer, washing hands, then we can remove that spread. Also, we can focus on custodial cleaning and remove it from, say, floors and commonly touched surfaces. If these surfaces are frequently disinfected, then we're removing that reservoir and removing that ability of that bacteria to spread from that surface to other patients. These are all considered universal prevention because they're not targeting a specific bacteria, they're targeting anything that lives there. We can also have targeted protection that focuses on specific organisms. So if we know that a patient has a particular type of infection that's antibiotic resistant, that patient can be isolated. We can look for mechanisms that specifically target that particular organism, and we can up protective equipment, such as with gowns, masks, and signs on doors to make sure that if there's an antibiotic resistant organism in this particular room, it is isolated from the rest of the patients to try and remove it from the population. However, so all of these preventions are currently in place and are very proactive about using them. However, even with all of these, we still see this rise in antibiotic-resistant infections. So it's still not 100% effective, and we still don't understand enough about what's going on in the environment to cause this spread. So for the rest of this presentation, I'm really going to focus on particularly contaminated surfaces. So I'm talking about door handles, sinks, toilets, bed rails, places that are commonly touched in the hospital. And we're really going to focus on what's there and how is it potentially spreading from one location to another, the transmission dynamics of these ARO reservoirs. Now, I'm going to set you up with two different hypotheses that are potentially happening and potentially that we see this spread and transmission dynamics. The first one is what I've kind of talked about before, that we have a patient source, right? A patient given antibiotics, then has antibiotic-resistant organisms in their gut. It transfers to surfaces and then transfers back to a patient. And this is, of course, a loop that can continue for as long as a hospital is in service. In this model, we expect that the organisms present are often human gut pathobionts. So pathobionts are things that could be a pathogen or could not be a pathogen, that they're commonly found in the gut and aren't always necessarily causing infections, but in a particular situation can become pathogenic and cause infections. So in this situation, we expect the organisms that we find to be related to the human gut and come from fecal contamination. And we expect the reservoirs that we're interested in to be these frequently touched surfaces where patients and visitors and healthcare workers are interacting with the environment. In our second model, so this is the main model that we've primarily been focusing on, and a lot of evidence have shown support for this, primarily in low to middle income countries, such as Pakistan and Singapore, where they've done surveys of the hospital environment and found a lot of different human gut related bacteria on surfaces to potentially spread. However, when we look in US hospitals, we don't often find necessarily these human gut pathobionts on surfaces, that different cleaning procedures are doing a really good job at removing them. And so this comes into our second model of environmental source. What we have found is that there have been outbreaks that are often specifically related to water sources. Say a contaminated water fountain in neonatal ICU, the NICU, spread to a dozen or so infections in this area, right? And so we find that the common source for this infection is actually infrastructure in this ICU. And that's these areas, especially water sources, are perfect places for bacteria to grow. They're moist, they're hard to clean, they're hard to get into. It's hard to assess that there's even bacteria in there, yet at the same time, they're able to travel outside of this infrastructure and onto, again, commonly touched surfaces onto healthcare workers, patients, and visitors, and then inevitably into patients who are susceptible and can get sick. Now, the issue with this is that the organisms are environmental pathobionts. When we think of environmental ones, no matter what surface you're going into, no matter what house, no matter what hospital, there will always be bacteria present. It's impossible to remove 100% of bacteria from everywhere. So it's more important to figure out which environmental bacteria are dangerous and which are dangerous to patients versus which are just natively there and impossible to get rid of under all circumstances. In this case, what we're looking at is environmental pathobionts, and our reservoir isn't necessarily the surfaces which are being thoroughly cleaned, but instead this hard to clean infrastructure. When things like this have been noted in other hospitals, often the only steps that can be taken to prevent transmission is to remove the infrastructure. So they would have to say, close down the ward, take out all the sinks and replace them with new sinks, or replace them with sinks that have, say, close in specific ways so that you can disinfect into the sink. So these are often much harder to deal with, but also create lasting problems. So these are the two general models of antibiotic-resistant organism reservoirs that I'm talking about, and I will come back to multiple times. All right, so the goal of this study then is to characterize transmission dynamics and to determine future interventions to improve hygiene and decrease transmission, to decrease ARO spread in hospitals and in ICUs. There are three main questions I'll be focusing on. What hospital environments are currently acting as reservoirs in this US hospital that we are surveying? When does reservoir colonization occur? And I'll talk a little bit more about how we can address that. And how dangerous are these bacteria? Are they actively infecting patients, or are they just always present? Can we find evidence of transmission to patients? All right. Now, there's a lot of potential places that we could do this study, and we are choosing to do it in the bone marrow transplant patient population. Bone marrow transplant, or BMT ICUs, are a good place for the study because the patients there are at the highest risk of infection. They have cancer, they have suppressed immune systems, they're on chemotherapy, and they're generally on a lot of antibiotics. This again creates a perfect storm for patients to become infected with environmental bacteria or other bacteria that is found on their surfaces. And they have the lowest risk of recovering from these bacteria when they do get an infection. So we will be focusing on this BMT ICU. Now, what areas did we swab? We looked at 13 different locations in this study. We looked at places that were frequently touched, such as the bedside rail, nurse call button, computer, light switch, gown and glove, and we looked at water-related places as well, such as sink drains and sink water. Also in our ICU, there is an attached bathroom on each ICU room, and from there we could look at a toilet, an in-room sofa, and a bathroom sink. So all of these are within the ICU room, and each of them were surveyed. Then we have another unique opportunity. So we didn't just do this in a ICU that has been established for a while. We did it in two ICUs. One, what I'll call the old ICU, has been established for decades and has been a BMT ICU for that long and has had a long time to establish different bacterial reservoirs. But we had a unique opportunity with the building of a new tower. This is at Barnes Jewish, where we did this survey. This new tower allowed us to look at kind of the natural history of a hospital. We could see what happens before patients even enter the building, when patients are first arrived, and then for a year upwards. And not only can we look at that new kind of natural history of the hospital, but we can compare it to the old ICU where the patients and healthcare workers all transferred from. So we have a great kind of control that looks at what did it look like, what's the differences in ARO establishment. Here's some pictures of our actual sampling, different locations. And we did this in 6 different ICU rooms. So we have multiple rooms of this BMT ICU for 13 different collection sites. I'll also mention, but we won't talk about too much, we did communal surfaces as well. So places that were not in the ICU room where visitors would be more likely to interact with. We did our sampling, we did longitudinal sampling. So we did it over the course of a year. First, we did in the old ICU. So each of these dots represent a sample time point. The big dots are for a month, and the small dumps are for 2-week intervals. So we have 3 sampling points in the old ICU, and then we switch over to the new. We start in this pink, which is before patients even enter the ICU, and then we have for up to a year of sampling when patients are in this new ICU. We collected 1,666 samples, mostly from ICU rooms. These are different swabs, but also communal swabs and patient stool. Now, patient stool is important because this is what we'd expect would be the potential material that is transferring these antibiotic-resistant organisms. We also looked at patient blood cultures. So when we have a blood culture, it means that patient was actively infected, had a blood infection with this bacteria, and that was the causative agent of infection. So we can see not only with the stool what they're spreading and shedding across the hospital, but also what is causing infections in these patients. So from the swabs, we did selective microbiological culture. This just means we selected specifically for antibiotic-resistant organisms. If we didn't, we would have gotten a ton of different bacteria, but we're not necessarily interested in everything that's there. We're interested in the stuff that's the most dangerous. We're interested in these things that are antibiotic-resistant, such as Pseudomonas, Enterococcus, Stenotrophomonas, Candida, etc. Then we kind of put this through the microbiology gauntlet. We used MALDI-TOF to identify species. We used AST to look at antibiotic susceptibility testing, and then we did DNA to do whole genome sequencing, which I'll talk a little bit more about this process in a few slides. MALDI-TOF just allows us straight from culture to use mass spectrometry to identify what a species is. And this is a growing database that if we take just a little bit of culture, we can tell you what the individual species and taxa of that species is, of that organism is. Antibiotic susceptibility testing was also done. So we know that we're selecting for antibiotic-resistant organisms using the different media, but we don't actually know phenotypically how resistant they are to different antibiotics. This assay allows us to, here's a lawn growth, so all blue is bacteria, and you put an antimicrobial disc on this growth. Where the bacteria is resistant, you will see, if it's resistant to this antibiotic, you'll see this big, wide circle of inhibition, right? So the bacteria can't grow right up to that disc. Where it is resistant, you'll see a small circle, so it can grow right up to that disc. That bacteria antibiotic is not decreasing its growth at all. So this way we can test a lot of different antibiotics and see what the actual phenotypic resistance is. All right, so on to results. What did we find? Well, from all of these different cultures, we found a lot of different antibiotic-resistant organisms, 746 to be exact. And where did we find them? Every single one of our BMT ICU rooms yielded antibiotic-resistant organisms. We found them all over the place. We found them in every single communal collection site. We found them in we found them in 12 of our 13 different sample locations. So we found them in sink drains, on bedside rails, on nurse call buttons, on computers. We didn't find them at every single time point, but we consistently found them on every location. This means these antibiotic-resistant organisms were all over the place. But they weren't all over the place equally. So here, we're looking at number of isolates per sample collection. So this is a metric that we can look at kind of microbial burden, how much bacteria was found in general on these different locations. And on the left, we're looking at the old ICU. On the right, we're looking at the new ICU for each of our different locations that were sampled. And these light gray bars just indicate that that was not sampled in the old ICU. The old ICU did not have the attached bathrooms, so we don't have a bathroom sink drain, we don't have a toilet. But the new had these locations, so we can compare both of them. And you can see, while we found bacteria in all of these locations, we found them primarily in sink drains. Sink drains had a much higher microbial burden, a significantly higher microbial burden than any of our other locations, by over double in each place, right? So this means that while we did find it on, like, say, the sofa or the bedside rail or the floor, we mostly found it in these sink drains, which here you can see are kind of gross. And you can imagine this is because while you can clean off the top of the sink drain, you can't necessarily clean within it very efficiently. And we stuck the swab down in there to swab as much of it as we could. So this is the place that we found the most bacteria. What types of bacteria were there? Well, we found a good number of all of the things that I mentioned before, all these different antibiotic resistance that you've seen on potentially the news. Pseudomonas aeruginosa, Enterobacteriaceae, Actinobacter, Stenotrophomonas, and we found them in particularly high amounts of Pseudomonas aeruginosa and Stenotrophomonas, which I'll go into a little bit later as we dig into what types of things were found. All right, so here we're looking at, well, where were these found, right? So we find things all over the place. We find all these different species, but we don't find an equal amount of species all over the place. So here we're looking at the MALDI identified, the mass spec identified genus for all the genus we find in both the old ICU and the new ICU. And we're looking at isolate count. What I mean by isolate is when we have, when we can separate a bacteria individually from that, right? So we find Pseudomonas more than any other genera in these ICU. And we primarily find it in these blue instances. So these are the in-room sink and the bathroom sink. Next we find a large amount of Stenotrophomonas, again, primarily in sink drains, in bathroom and in-room sink drains. After that, we find kind of fewer amounts of everything, and often potentially just in patients or just in environmental samples. We'll dig into a little bit deeper what's going on at this level later on. So now, when did we find these things? Right, so here's our timeline again. We have the old ICU, the new ICU, and pink for before patients entered. Every single time point that we sampled yielded antibiotic-resistant organisms. So this includes before patients even entered the ICU. This is before after they finished the building, after they'd done all the final terminal cleaning for construction. Once we sampled, there were still antibiotic-resistant organisms before patients had even entered. When we look at how much, so here we're looking at number of isolates, so how many bacterial isolates we find over sink drain sample collections by collections that we took. And here it's matched up with our timeline. And we find that the ones in the old ICU, we have a higher microbial burden. We find more isolates in sink drains in the old ICU, but when we look at the new, we find a fairly steady level of isolates. And this includes before patients even move in, right? So we find significantly more in the old ICU, but in the new ICU, they're before patients move in and there's no increase. So what does this mean for our hypotheses that we're looking at, right? If we expect these antibiotic-resistant organisms to be coming from patients, then we would expect there to be a lower rate before patients moved in. If they're coming from patients, how are they present before patients even get there? And we would expect potentially an increase over time as the patients inhabit the ICU, as they shed more antibiotic-resistant organisms, and as this cycle continues, we would expect an increase. We don't see either of these things. We see the bacteria are present before patients move in, and we see a very steady rate of collection for this entire year. This suggests that they are not potentially coming just from patients, that instead we're seeing something that's environmentally linked, something that's present from day one before that hospital even opened up. And this is true, so before we were just looking at sink drains, this is true for other surface sample collection as well, although we see that these rates of collection are much lower and it's highly variable. But in general, we see a similar rates from before patients move in all the way up to a year, with some instances of very high microbial burden. So going back to our goals of characterizing this, what does this mean for our three different questions? Well, what hospital environments are acting as reservoirs, and when does reservoir colonization occur? We find that sink drains in particular have a high microbial burden over time. The rest of the surfaces seem like they're being cleaned well and don't necessarily have very high microbial burden, although we do find microbes on them over time. But sink drains in particular seem to be a place where we have this reservoir forming, where bacteria stay and remain for the entire year of collection. And how about when does reservoir colonization occur? Well, this we're going to look into a little bit more, but we find that AROs are present before patients even move into the ICU. So even sampling after construction isn't soon enough to let us know when actually reservoir colonization is occurring. And as for how much these environmental bacteria are affecting patients, that we'll have to get into a little bit more. Right now, I've looked at it just as an aggregate level. I've been looking at antibiotic-resistant organisms as a whole and not looking at specific species. But we should remember that each of these different organisms can be very different and cause very different types of infections and have very different types of transmission patterns. So we can't just look at it as a whole. And I think we need to look at it not just on a species level, but instead get down to a strain level. Can we actually track strains and look at where an individual strain is going over time? So we did this using whole genome sequencing, which allows strain tracking. Whole genome sequencing, what we did was shotgun sequencing. So we took an organism, took the bacteria, break up their cell wall, extract all the DNA. We chop up that DNA into little fragments and send it all through the sequencer. Then we take all these little fragments that we get back from the sequencer and put them together to create a genome assembly so that we know what each of these organisms' genomes look like. From this genome assembly, we can do all sorts of different analyses, including gene annotation, to know what kind of genes are present in each of these different organisms. This allows us to do really in-depth species identification. There's a lot of different ways to identify species, but whole genomes allows the best kind of grasp of species and bacteria. And rather than think of all the different types of species concepts, we just have a cutoff of when we have two different genomes aligned, average nucleotide identity has to be greater than 96%. If this is true, we're looking at the same species. If it's not true, we're looking at different species. And here I'll just kind of remind you of our taxonomic hierarchy, right? So when I'm talking about species, well, I'll mention family at some level. And then we were previously talking about genus. And so now we're getting down to the species level with identification. And then a little bit about subspecies, but I'm actually going to talk about more of a strain level than a subspecies in bacteria. So whole genomes allow us, yep.
Just real quick, we're at 30 minutes.
Thank you very much. So this strain identification allows us to, so whole genome sequencing allows us to go into strain identification using multi-locus strain typing. So this way we can actually extract a few genes that we're interested in, look at the alleles of these genes, and figure out what strain type they are. This is helpful because it allows us to compare strains across the globe, as well as focus in on these individual strains. We wouldn't necessarily be surprised to find the same species in these different locations. However, if it's the same strain, this is implying that we can actually track individual strains across location, that there is some kind of clonal reproduction happening, and that the same bacteria that is found in the sink is the same bacteria that we find in a patient, is the same bacteria that we find in the sink over time. To get to that level, we need to actually look at strain levels instead of just species levels. We can also do phylogenetic analyses and antibiotic-resistant genes when we're looking at whole genome sequencing. But I'm really going to focus on this difference between genus level, species level, and our strain tracking, which I'll do through MLST. So we're looking at whole genome sequencing of 3 different taxa. I'm going to focus on Enterobacteriaceae. This is a family level. And the bacteria in this family, while highly diverse, the antibiotic-resistant organisms of them typically are associated with the human gut, are frequently associated with the human gut, among other things. And so these would be the things that we'd expect to be human gut pathobionts and potentially in fecal contamination. We're also going to look at Stenotrophomonas and Pseudomonas. These are things that cause human infections, but are also frequently associated with water sources and are found in sinks and sink drains, even in places that are not hospitals, even in places where we don't see antibiotic-resistant infections. So let's start with Enterobacteriaceae. That is particularly associated with fecal contamination. This is what we'd expect in our Model 1, our patient source bacteria. We find a lot of different bacteria of Enterobacteriaceae, but they are primarily found here. Here we have a list of all the different species found, as well as our isolate count. They are primarily found in blood and stool, in the red and brown color. We see very little blues, and we see very little blacks. What does this mean? It means that we find these bacteria primarily associated with patients and very, very rarely on surfaces. So they're not necessarily contaminating many surfaces that we see. Here, we're tracking individual species across time and space. So we have a number of different locations where these bacteria are found, and then all the weeks of our collection. And how many different Enterobacteriaceae did we find on surfaces? We found 7 total. So 7 instances. They were never, except in a single case where we find it on top of each other, they were always different species, which means they are not the same strain. They are not creating reservoirs. They are not being found across time and space. There's no spreading, right? We are finding individual instances of contamination by Enterobacteriaceae, but it doesn't look like a giant reservoir problem that's happening in the hospital. We're going to focus in on particularly this little green dot here of the E. coli. When we do MLST tracking, so this is looking at strain typing of this particular E. coli, we do find it in stool and blood. So this single instance could be an evidence of transfer from a patient, stool or blood, onto a surface. But we find no evidence from surfaces into stool and blood. And we don't even find in that same week a patient that had it. So this generally supports the idea that while there might be evidence of patients depositing a bacteria onto surfaces, we don't see a lot of transfer back. This model is not necessarily well supported, model 1. So let's look at our environmental things, starting with Stenotrophomonas, which is often considered a water source. Here, we find Stenotrophomonas in sink drains every single week besides the first week that we did sampling, in variable amounts. What I have in red bars here is when we find human infections with Stenotrophomonas. And it happens later on in the hospital development. However, when we look at the species, so this is looking at the genus level of Stenotrophomonas, when we look at the individual species, we find no overlap between any patient samples and environmental sample species, right? So Stenotrophospeltophilia, which was the most found species, has no blood or stool samples. It is only found on surfaces. When we track strain types, again, we're looking at MLST. Here's all of the different strains in different colors. We find that there are, particularly let's look at this one in light blue, is found all the way in a sink drain from before patients arrive all the way to 61 weeks after. This would suggest a reservoir is formed, and this reservoir is consistently there the entire time the hospital has been open. But it never transfers into patient blood or patient stool, which is a good thing. Finally, we'll look at Pseudomonas aeruginosa. Pseudomonas aeruginosa, again, is found across all weeks, and we primarily just find Pseudomonas aeruginosa of all the Pseudomonas that we surveyed. However, here, when we look at strain typing, we find a very different pattern that's happening. Let's look at this yellow one. It starts before patients move in, and it's found all the way up to 61 weeks, so the entire time it was here, and it spreads. It starts out in one room and ends up in another, and another, and another, until it's found in all 6 rooms that we surveyed. Only in the new ICU, never found in the old. And now what's really unfortunate is here when we look at the blood cultures, way at the bottom. We find evidence of the same strain in patients, which means even though it's found initially in the environment and is spread across these ICU, it is directly transferring into patients as well and potentially causing these infections. And we find it before the patients even arrive in there, which suggests that they're getting it from the environment and not necessarily the other way around. This is a lot of support then for our environmental source hypothesis, that these sink drains are acting as reservoirs with environmental bacteria that can then be transferred potentially to other surfaces, but ultimately to patients and causing patient infections. So for our goals, yes, AROs are present in the ICU before it's open, sink drains are acting as reservoirs, and when we look at Pseudomonas aeruginosa, we find reservoirs that last over a year in these sink drains and infect patients. So this makes us rethink our general model for the spread of AROs in the ICU. We can't just focus on things that are patient spread and patient origin, because here we find very little evidence of patient contamination, but instead we find things that are present environmentally in the ICU before patients arise that are spreading across the ICU and infecting patients. If we want a method to decrease transmission, we have to focus not necessarily on fecal contamination from patients, but on how to clean out these sink drains and remove the bacterial reservoirs that are inhabiting those areas. So all of this will come to detect and eliminate reservoirs of antibiotic-resistant organisms and create safer hospitals with fewer antibiotic resistance transmission. I'd like to acknowledge my lab and everyone who's helped me out with this again, and I am happy to take any questions. Oh, can't hear you, Shelley.
We do have another number of questions that have come in, so I appreciate you giving us time
Awesome.
to talk about these. So let me walk through some of them. All right, so we have some kind of basic research questions, and then we have some that are more specific to your actual research. So some questions about which of these antibiotic resistant organisms would you find to be most harmful to humans?
Yes, so that is a difficult question because it kind of comes down to how many infections we find and how frequently it infects, and there's all sorts of different types of infections. So while the CDC does have levels of threats, so things like carbapenemase resistance, Enterobacteriaceae are often considered at the top of the list of worst antibiotic resistant organisms. Why? Because they're found all over the place and they're resistant to carbapenemases, which are often our last line of defense antibiotics. So while they're not all created equal and they have all sorts of different infections, and you will have to consider that on how dangerous they are, things that are resistant to their last line antibiotics are the things that really we want to focus on getting rid of because those we have very little treatment for. Other things that are resistant to say one or two different antibiotics are dangerous and something to keep an eye on, but they're maybe not, they're still treatable by using a different antibiotic treatment.
Thank you. I have some really basic questions here. So just asking questions about how do you sample? So when you're sampling, what are you doing to make sure you have an accurate sample of your bacteria?
Yep. So here we want an accurate sample and we want to be consistent as possible. We use the eSwab kit. eSwab is a marketed kit that does a really good job of preserving the bacteria as soon as we get it into its liquid, and then we can immediately kind of transfer it over to the microbiology lab so we can start culturing it, right? So no process in this did we freeze it. We didn't do anything to kill the cells. We tried to collect as many things as we possibly could. To be consistent with it, we took 3 swabs from each of these kits and swabbed across the entire surface area in a very consistent manner to make sure we're always collecting the same surfaces. And then those 3 different replicates were then stored and then sent off to the microbiology lab.
Thank you. Some questions here about COVID. I know that we were doing this experiment during a specific amount of time, but did you test during COVID at all, or have you seen any changes or think there might be?
So this analysis, these sampling were done in 2017, 2018. So long before we had any COVID exposures. However, we are doing currently a follow-up project from this where we're actually looking at intervention studies to clean out those sink drains. So we are actively working on the next step of fixing this problem. And that started before COVID started. And then our sampling got interrupted and then COVID started. And all of a sudden we have all sorts of different precautions to take. Now, I think it's going to be really fascinating to look at that data and look at how COVID and different precautionary measures have changed what we find. With our different microbiology, biological selective culturing, we're not actually looking for viruses. So we won't be able to culture any COVID and see what that looks like. But you can imagine that the different patterns of antibiotic resistance are going to dramatically change, especially as COVID has resulted in a lot of people getting more antibiotics than they would have, which the virus does not respond to antibiotics. However, you often see an increase in antibiotic usage during the COVID pandemic. So we should potentially even see interaction there. I think it'll be really fascinating to look deeper into.
That's exciting just to have this opportunity come into play while you're in the middle of a research project. That's pretty great. We have a question, a specific question. It says strain 1894 is not present in the environment when it is present in a patient. And why might that be?
So so here, I think it's a little bit with how our sampling works. While we are extensively sampling with these swabs, there are going to be times where we potentially don't sample something that is still there, right? Especially because we are only selecting an individual colony each time from these plates. We want isolates, so we want to make sure we're getting only an individual colony and not mixed colonies. But the downside of this means that we are missing some of the diversity. So maybe we
Mm,
picked a different strain rather than the strain we're interested in. And
mm,
each time we've got kind of a mixed community of different strains present. So I would say just because we don't find a strain at any given time point does not mean it was necessarily not there over that time.
oh, thank you. Greg, do you want to ask your question?
I could find the unmute, yes. So I'm sorry if I missed it. It was way back in the beginning. Did you swab the air vents at all to look at ventilation? Or
Nope, we
okay,
didn't look at any ventilation, so we didn't do any air vents, anything related to that. But I think that would be another cool question. I think especially when we mentioned with COVID as well, like because I think things changed specifically with the different pandemics. So
overnight.
I feel like that'd be a whole new study to really focus on.
Neat.
Very cool.
Thank you. Um, so, so I have a question again, kind of like a linking things together question. So, what are those the particular situations where the gut-related bacteria or infections become pathogens?
Yes, so this is something that it's a really big question that my lab is focused on a lot because it's not always very clear. So
Mm,
in every single human, we have a lot of, say, Enterobacteriaceae and gut-associated pathogens or pathobionts in our stomach. And usually they are not dangerous. They are things that are actually commensal and potentially help you across your life. It's important to have a healthy gut bacteria. However, we find instances where these bacteria that are happy commensals become pathogenic, can potentially cross over the gut barrier, end up in the bloodstream, and cause an infection. Sometimes it's internally related, that you end up infected by something that was in your gut. And sometimes we see it from kind of these outside loops, right? Where we see fecal contamination that ends up in a different patient and causes an infection in that patient. I
mm,
think I'll specifically mention that these are patients that are incredibly susceptible to antibiotic or to infections in general, right?
mm,
So maybe these would be happy commensal bacteria in normal healthy humans, but all of a sudden when your immune system is suppressed, when you're on a lot of different drugs, chemotherapy, all of a sudden
mm.
something that was typically commensal now causes an infection in ill individual.
Thank you. So we're working mostly in hospitals. Get some questions about things that are happening outside of hospitals. So how likely would it be to find similar results in areas like mall restrooms and restaurants and places that aren't cleaned quite as thoroughly with such intensive cleaning
Yep.
supplies?
So I would not be surprised if in malls and other locations, even houses, that sink drains are going to be persistently an area for bacterial biofilm development, right? They're moist, they're hard to clean, it's hard to get into those pipes, and these are perfect locations for things especially that are like water environmental bugs to live and thrive and not be disturbed over time. What we might not find though is actually the antibiotic resistance. So antibiotic resistance usually is selected for in the presence of heavy use of antibiotics. So in ICUs, this makes a
Mm,
lot of sense because patients are given them and thus they become more antibiotic resistant. So I'd hope that in malls and other places, while we might find more bacteria, especially on surfaces, they might not be the worst ones that we're talking about in the ICU.
yeah, thank you. So I have questions about what do we do about this? And so one of the questions was, what's your opinion on using other organisms, things like TSO worms, to try to get rid of these harmful bacteria?
So I love the creativity of thought in that, and I think it's a really cool, special mental exercise, something we should be doing in labs to test out. I don't know much about the worms in particular, but we have looked at different kind of probiotic solutions, right?
Mm,
Using bacteria to fight bacteria, and if we can get some really good, especially like competitive or different interactive things going on, you might be able to exclude these really dangerous pathobionts. Um, However, I think it's really important to understand what's going on before you introduce it into an environment where these patients are very sick, and maybe you might cause a worse infection from something that's supposed to be potentially fighting your other bacteria. But I think it's a really cool idea.
Thank you. Are there other kinds of like specialty cleaning that works well in these situations?
Yeah, so I mean, this has been an evolving process for decades, right? Where we find that, and I think our hospital RCU is in many ways a success story. They are doing a great job cleaning off surfaces. I really want to emphasize that because we don't find many gut associated or most like any bacteria at all on these surfaces. So when things pop up, they're almost immediately wiped away on bedside rails, on you know, on light switches, on everything that the patients are touching. So I think our typical cleaning methods are doing a pretty good job. What we need to focus on is better cleaning methods for places that are less easy to clean, such as these sink drains. And in this new study that I talked about, we're looking at intervention methods. Now, sink drains are hard because you can't just pour bleach down a sink drain. You can, but if it doesn't touch the bacteria, if it doesn't like rest on that surface for a while, you're not actually going to clean anything out. So we're looking for methods with like different foam things, different valves we can shut that would close up the sink, allow us to thoroughly disinfect all of the bacteria that are there, and then rinse it all away. So I think the new methods of cleaning will have to really focus on what do we do for places that are hard to reach and hard to routinely clean. And some of that will be in like new infrastructure. Some of it will be in we need to find methods that get into that infrastructure we already have. We can't rebuild every single hospital we have.
Yeah, thank you. So in the cleaning methods that they're using currently that are doing a good job, is this is not antibiotic soap? Is this more like bleach and things that actually kill the cells themselves?
Yep, so a lot of it is bleach, which does a great job at, killing absolutely everything. A lot of the methods of cleaning, though, is not necessarily the best structured. It's often like nurses wiping down when they have the time, and then custodials coming in every so often. Again, the main focus is keeping the patient healthy and happy, and that often doesn't necessarily fall in well with a full custodial staff coming in every single, hour, couple hours to clean off everything.
Thank you. This has been wonderful. We are really interested. We're having a bunch of questions about your career trajectory, and I was wondering if you could share about your path that you've taken to get to this point.
Yeah, I'd be happy to. So let's see. Well, I'll start out way back out of high school. I was interested in biology, and I went to a small liberal arts college, Gustavus Adolphus in Minnesota, where I got to really explore kind of a bunch of different, like I was interested in biology, chemistry, biochemistry, physics, like a lot of different science routes, but not really sure what I specifically wanted to do. And I came from a family that doesn't have a lot of history in higher education, right? So we didn't know that a PhD would be an option. It's something that never even came up. It was okay, you'll finish college and then, move off onto your career. So I really had a great opportunity. It was my first year, but fairly early on, to just try out a little bit of research. It was in a classroom environment, so it wasn't an independent research project, but it was enough to kind of get me thinking about what research looks like. And this was with yeast and different genomic kind of aspects of yeast. And I think that really got me thinking of, okay, well, what different opportunities are there? And from there, I looked for summer opportunities. So I would recommend students to really keep an eye out across the country for different fellowships and scholarships to apply for. They'll let you do summer research. I found a summer research program that was at Washington University that I actually applied to and did an internship that would, pay for, well, one, give me a stipend for the entire time I was doing research. It paid for housing, paid for my travel out there, and I got to spend 8 weeks doing research in a lab for a short internship to really dive into the research. And I came away from that having a much broader grasp of what research is like, as well as understanding more about like the PhD process and kind of what goes on if you continue to stay in academia research. So this really inspired me, and I wasn't really sure what kind of research I wanted to do, but I started looking for more and more opportunities, both at my college as well as colleges across the country. Um, and I was lucky to really find some different kind of research that I enjoyed. I specifically was interested in evolution ecology, which is not really, I mean, I think it's important to note that there's evolution ecology research in everything. So right now I'm doing very medical, clinically focused research. But even here, we're looking at different species concepts. We're looking at the evolution of antibiotic resistance in selective pressures and how it impacts natural histories of building. These are all things that really were established from an evolution ecology background. But so from there, I was interested in doing research on actually brains in electric fish. So something really different from what I'm doing now. And I spent my PhD at WashU really focusing in on brain size evolution. We did a lot of, got a lot of cool fish in lab, did a lot of behavioral assays and things like that to look at kind of the neuroscience of these different fish. And I really enjoyed that research. But when I finished my PhD, I was interested in something that was more broadly applicable, something that would really like still keep with my evolution ecology interests, but also like maybe potentially be really helpful in a clinical environment. So I looked up for postdocs and, you know, sent a couple emails, looked around for people who were doing the kind of research that I was interested in. And that's how I ended up in the current position I'm at with postdoctoral research. A postdoc is often a training kind of position where you spend a couple years potentially doing a postdoc, building up your resume of different papers that you've written and really focusing on the next step and looking for different positions after that. And I'm happy to say I actually have a new position that I'll be transitioning into this summer where I'm going to be an instructor faculty at Washington University. So this is a research position and I'll be on the research track to being my own primary investigator and doing clinically related research.
Wow, congratulations. That is
Thank you.
awesome.
Thank you.
That is so wonderful. Thank you for sharing your path. That's really fascinating how you didn't go in one direct line toward where you're at now. You really moved around and thought about different things. I appreciate that. And
There's kind of always time to kind of like change your interests and really kind of like focus in on different things. And I find that it often builds. Like if you find like after a few years you're interested in something else, like I'm taking with me my evolution ecology experience. Even the neuroscience stuff I did with brains. While this project I focused on didn't involve brains at all, I'm also doing stuff that looks at neurodegenerative disease in HIV and how the microbiome is impacting, you know, brains in these patients. So I feel like
wow,
there's still a lot of ways to use things that you've used in the past and continue to grow and develop. And it's always fun to learn new things too and keep building.
that's exciting. Thank you. Thank you for also sharing that you came from a family that didn't necessarily think, oh, higher ed, because a lot of our students are coming from that perspective. So I really appreciate you sharing that piece. So I have a number of questions that have come in from students and just various things. And you've kind of mentioned some ideas about getting into research early and some questions about, you know, what would you recommend to actually find those programs?
Yes. So let's see. I know that there are that's kind of tough because there's a lot of different things and it really depends on what you're interested in and which program you want to go into. But I wouldn't be afraid to do broad searches for any type of research program and any type of funding. You can even look for at a national level, there's often like individual fellowships you can apply for that will help pay for individual experiences, and then you can go to potential places that you're interested in. Well, it's always easier to start locally and start talking to your individual professors that are around from their experiences and what they have open. I wouldn't be afraid to branch out from that as well too and start looking at larger universities because I liked Gustavus Adolphus and it was really good for different internship and research experiences. They often didn't have as big a program. They didn't have any of the kind of biomedical and clinical related work. So if I was interested in that, it was good to hit up these different schools and see what funding they have available. It's usually where I start. And then what labs are interested, what labs are available that I'd be interested in working in.
I kind of want
Thank
to,
you.
can I piggyback and add a little bit just because I've had so many conversations with Kim and I'm like, she's left out details that would be helpful. So I think, so Kim is very social and Kim is very good at talking to people. And I would argue that that's really helped her a lot. And I think as undergrads, it can be intimidating to talk to professors. But the earlier that you talk to people and the more you talk to people, the more opportunities will open up. So some of the details that Dr. Sukum, I'll casually call her Kim, but some of the things that Dr. Sukum left out are that her summer internship that she had when she was an undergrad was at the Missouri Botanical Garden. So she met people at WashU during that time. So when it came to apply, she already kind of knew some people, which was kind of nice.
Mhm.
And at WashU, we have rotations, which means when you arrive, you're not set to work in a particular lab yet. So Kim actually explored a lot and she worked in several different labs until she found the one that was the right fit for her.
Yeah.
And then her postdoc lab was actually one of our other friends' PhD labs. And so Kim met her friend's PhD advisor, made those connections, and then that led to her next position. And so
Yes.
having those conversations, even if they're casual, but just making those connections and building those bridges can lead to things that you're not even aware of later. And I think her current position is what has led her into her next upcoming job. So
Yeah, yeah. Sarah knows it well. And I would totally recommend, even if someone like doesn't necessarily have room in their lab or room at this time, like learning more about their research if you're interested in it, learning more about like that field and what people, because yeah, I think Sarah was right. It's a lot of networking, it's a lot of kind of interacting with individuals and learning about opportunities that way.
Thank you. Yeah, that was actually several questions that you just answered between the two of you. So thank you. So I have some questions here about just thinking about your career so far and excuse me, what kind of what was the most impressive part or the most memorable part of your career so far? Like thinking about what really changed things for you.
Ooh. Um, I feel like there have been a bunch of pivotal points in my
Mm.
career that really have gotten me thinking in different ways and really kind of like broadened my scopes. One of them, I remember being an undergrad and going to a seminar on research. Actually, no, sorry, this was a grad school, and going to a seminar of something that I wasn't necessarily, I didn't think I'd be that interested in it. It was actually about bacteria. I was working on, fish at the time. Completely different stuff, in no way related to the things I was currently working on, or even necessarily things that I thought I'd be interested in. But I took the opportunity to go to the seminar and sit down and hear someone new talk. And I remember it was just kind of this mind-blowing experience learning about antibiotic-resistant organisms, where I'm sitting there thinking the research is really cool. It's stuff that I had no idea about, and it seemed like there was a lot of holes, right? In a good way, in a like, I really want to fill that hole. I really want to learn more about that particular thing and help kind of bridge that gap. So yeah, I feel like that was a really pivotal point in my grad school where I started thinking, you know what? Maybe not just fish brains. Maybe there's some other things that are really cool and I'm really interested in doing. Um, I feel like those kind of moments tend to be really pivotal. Also, there's been a lot of kind of like individual research. So research day to day, I don't know if anyone has experience on this. It often you kind of get really kind of buried in the day to day of it, right? So imagine in my project, doing swabs every week is not really fun. When you do the swabs, you're not really thinking about what you're picking up. You're just going to these different places, swabbing it, putting it in the liquid, sending it off, and some other part of the team is doing different things, and that is harder to see what the actual science is coming from it. And it's easy to get kind of buried in those details. And so it's always fun when you go from that to kind of more of the analysis, when you get to take that next step back and see, okay, here's what my year's worth of sampling looks like. What did we find? What kind of patterns are we showing? What kind of story is there? And there's definitely been a lot of times where, a project that you think, man, I don't think this went anywhere. I don't think, the cool things came from this. All of a sudden, you look at it in a different light, in a different kind of story environment, and it grows into something big and exciting.
Thank you. So I have several questions about you being one of the first people in your family to be going into higher ed. And
Mhm.
so one of the questions was, I think that I know a lot of my students feel is the pressure. Did you feel pressure to go into a specific career path because you were one of the first people? And what did you do with that?
Yeah, mine was more, my family was very excited for me to have gotten into college. So I very much took the initiative to look up for colleges and decide where I want to go, pick one and go there. And while my parents were very much that, they were interested in, the idea of potentially me going on to be a nurse or a doctor or something in one of these kind of tracks, um, they weren't really aware of the different options that were out there, right? It was
Mm.
very much, okay, these are professions that they've known, that they see around, and that they would love for me to continue. So while those were the things that came up because they knew it, when I started learning about research, when I started learning about the PhD and different options, it was kind of a little bit on me to show them that there is a bigger world out there. And they were very enthusiastic once they heard about it. They didn't realize a PhD was an option. They didn't know what research looks like or what, what does someone with their doctorate do. Um, but you can really kind of flip the story a bit when you think of, okay, well, doctors help a lot of individual lives. When you're doing research, when you do things like, so say my individual project might evolve into interventions that clean out those sinks and might save hundreds of lives at this hospital by preventing antibiotic-resistant organisms. Not just this, but maybe these principles will be used across multiple hospitals across the country, across the globe, and we can really start cleaning and pushing back on antibiotic-resistant organisms in some way. And so now, if your passion, if your interest is helping individual people, you can do it on more than just a doctor-patient level, more than just a nurse-patient level. You can do it on a research global level as well. So once I kind of showed my parents these different options, it also helps that a lot of the science research internships were paid to do work for the summer. So it wasn't okay, do this or do that. It's like, well, I can have a job and do research. And then when you went to my PhD, that also was paid. So after undergrad, I wasn't having to take up like, say a master's or something where you end up paying all of it. The PhD work actually had 6 years where I got a stipend, where I was doing my studies, getting my PhD, but also getting paid a reasonable amount the entire time as well.
Nice. Yeah, that's wonderful. I think that leads right into another question, just about work-life balance. Like, how have you been able to balance that time for your private life and time for your professional life throughout your career as undergrad and as a graduate and then as your postdoc?
Yes, so I think work-life balance is so important. And it's something that I've focused on not just where I'm currently at, but also in grad school and in my postdoc life. And it's not necessarily um, an easy thing. So with academia and with research, it often tends to be really stop and go. So you'll have times that are very busy where you're trying to get something done, and then you'll have kind of down times. I think it's really important for you as an individual to both understand that there's different flow periods, but also to set kind of hard limits. That, okay, I like work, I'm getting really caught up in it. It's really easy to take it home with you, do it whenever you want. But you also have to set times where, okay, I'm going home right now. I'm going back to my family. I'm not working on this anymore. And what I've found is that in my life, I've been pretty strict about making sure those limits are set up, and it's worked out really nicely. That I I have a good work-life balance. I have a lot of time with my family. I have a lot of time with social interactions, with friends, with weekends that don't involve just work. But it's easy to do the other as well.
Yeah, thank you. Thank you for sharing that. It's such an important concept for students and thinking about that as you're going into your professional career to really focus on making sure that you have those social and family times to not get overwhelmed with work. And I think one final question uh, to for me to ask is, when you're moving into this new career, which I'm so excited for you that you were able to get, do you think you'll continue to focus on the things that you've done so far, or are there totally new areas that you might be exploring?
So a little bit of both, but a lot of focus. So currently, I have a lot of different projects that span working on farm manure to look at the biology of that, to looking at brains and neurodegenerative disorder, to looking at surface contamination. So I've got a lot of projects all over the board. This
Wow.
new career opportunity allows me to actually really focus on particularly the clinical pathogen interactions, and I'm going to help develop. So I'm moving from primarily focusing on the research to this new position will split research with a more directorial role of building a pathogenomic center where we can bring together a bunch of different labs, a bunch of different kind of researchers, both at a graduate level and undergraduate level, and at a postdoctoral level, who will all focus on these different kind of collaborations and these different clinically relevant projects. So I'm going to continue research that I'm interested in, such as the hospital microbiome, but also really focus on like building infrastructure that will help more research, more collaboration, more interactions between researchers.
Wonderful. That's so exciting. I'm just thrilled for you and about the prospects of this.
Thank you.
Wonderful. I think that we're going to end with our questions for right now. and move on to just talking about a few other things. So I'm really excited that we had you as our final presenter for the this semester's biological seminar series. So thank you so much for sharing with us your work and all that you've done. This was really exciting, and I appreciate all the effort that you put into this this time. Everyone else who is here, I'd like to bring your attention to the Q&A. There's a feedback request for next semester that Dr. Eaton posted. There's a link there, and if you would be interested in having, we're starting to plan for fall and spring semester of next year. So if you know of other people that you would be interested in having here for the seminar series, please fill out that form, and we would love to hear back from you. And just also note that I will be sending out an email to everyone, all of our different majors, again, with that same kind of link, just to get your feedback, because we're really interested in finding speakers that you would be interested in hearing here in our seminar series in the fall. So thank you so much, Dr. Sukham. We really appreciate you being here today. And thank you to everyone who's been here. Dr. Sukham, if you could stick around for just a minute, Dr. Eaton and I will chat with you after this. Thank you. See you, everyone.
Good job.